Hi, welcome to the Bioinformatics Chat. Today we'll be talking about CRISPR. So, you know that CRISPR can be used to knock out or edit genes, but it's not a precise text editor where you can delete precisely some letters. It can result in all sorts of mutations. And today we'll be talking to Seung Woo Cho and HoJoon Lee, who designed a program to analyze those mutations. Seung Woo and HoJoon, welcome to the podcast.
Thank you.
Thank you for your invitation.
I'm curious to know about your personal backgrounds and how you got to study or work with CRISPR. Seung Woo, let's start with you.
My background is actually developing the genome engineering technology. So, like 6 years ago, I was one of the world's first person to show the CRISPR can be used for genome engineering. So we showed that the CRISPR can edit the eukaryotic genome, including the humans. And then I came to Stanford to study the human genome functions using CRISPR.
Cool. HoJoon, what about you?
So my background is bioinformatics, especially in cancer genomics. So analyzing patient data, sequencing data to decide which treatment option is best for them or what is their prognosis and diagnosis and so on. So kind of it's precision medicine or personalized medicine. So I like to analyzing all the sequencing data in many different ways. And soon when I work in the same building in Stanford. So we talk each other a lot, and then Seung is complaining about insertion, deletion, calling at the CRISPR-Cas9 sites and so on. Okay, maybe I can help you out. That's how we end up analyzing CRISPR. I'm usually analyzing whole exome, whole genome, cancer patient data, but this is my first time to analyze CRISPR-Cas9 mutation. But my specialty is actually in the variant calling in any sequencing data. So it was my perfect alternative of other application, interesting application.
Right. So Seung Woo, you study CRISPR in general, like all things related to CRISPR, or do you have some specific goal that you're trying to achieve?
For now, for example, the CRISPR have some limitations, like the CRISPR cannot edit or targetable sequences, or it has some variable efficiencies and off-target issues or any other issues. So I'm working to develop and engineer the CRISPR to improve their performance better. And then using the CRISPR, I'm trying to study the genomic function and also the epigenomic function of the human disease or other life.
I was curious, so in your paper, in the competing interests section, you say that you're preparing to file for some sort of intellectual property based on your work. Can you talk more about it?
So I think we can provide this software to academia or nonprofit research institute without any charge. But if some company using CRISPR technology want to use our program, I think they have to pay for us.
Okay. Yeah, I'm very curious how it works out for you, especially since you described in a lot of detail in your paper how your program works, and hopefully we'll talk about some of that today, so anyone could just re-implement it themselves, right? So walk us through a typical CRISPR experiment. What are the steps you need to do if you want to edit or knock out some genes?
First, you have to design the guide RNA sequence for your given target sequence, target genes, and then deliver the expression vector of Cas9 and guide RNA into the cells. Then all of the CRISPR constructs are expressed from the construct, then they can edit the genome. Usually it takes 2 to 3 days, or if you deliver the the RNP complex, you can see the mutations within 6 hours. And then we have to see and analyze the mutations because we cannot see the genomic sequence using the microscope or whatever. We cannot see actually. So usually now people using the high throughput sequencing, so purify the genomic DNAs from the cells and then amplify the targeted region using PCR, then make the sequencing library also using the PCR, and then just the sequencing. Then the sequencing machine provide the sequencing information just written by AT, GC, whatever, and then we have to analyze the sequencing data, what they mean. So our program is about the last step.
Yeah, I think it's a very good overview of the whole thing. And I wanted to stop on each part separately. So we start with designing the guide RNA. What are the rules there? So you know the exact genomic location, right? Sort of between two nucleotides where you want to introduce the double-strand break. And how do you go from there, from knowing the genomic location to the exact sequence of your guide RNA?
Right. So we should know the genomic sequence of the target region, and then the CRISPR has the PAM sequence is recognized by the Cas9 protein rather than the guide RNA. So most CRISPR systems have their unique PAM sequence. For example, the SP-Cas9 have recognized the dinucleotide of guanine. So first we search the dinucleotide of guanine sequence in the target region. And then most importantly, we have to search the target cover sequence in the genome. How many sequences similar to the target cover sequence? Because the CRISPR cannot distinguish the one or two different sequence from out of 20 target sequence. So we have to exclude the target sequence have some similar sequence in the entire genome. And then we just try.
Right. So you start by looking for this double G, for example, PAM sequence, and then from there you take some more nucleotides, right? Yeah.
Right.
How many do you need for the whole guide RNA?
It depends on the Cas9 type, but the most widely used CRISPR SP-Cas9 recognize the 20 nucleotides.
Right. And so that's it. You just need to synthesize this 22 nucleotide DNA,
right.
or and then you transcribe it to RNA.
The guide RNA is transcribed from the usually people using the U6 promoter and some backbone sequence around 80 nucleotides, and then put the 20 nucleotide into the vector. So usually it requires 24 or 25 oligonucleotides for cloning the vector.
Mm-hmm. And so then you have, as you said, you have to deliver this to the cells. So what kind of vector do you guys use?
We usually use the just normal U6 and CMV construct, the human expression vector, or sometimes we can use lentiviral vectors, whatever the DNA vector can harbor the Cas9 encoding sequence and guide RNA encoding sequence.
So can you talk in a bit more detail how that vector works, the standard human expression vector, what it is and how it works?
The CRISPR is composed of two different components. The first one is Cas9 protein, and the second one is guide RNA. So the Cas9 protein is expressed from any appropriate promoters, and it requires some enhancers and terminators. So people usually using the expression vector for human cells, like CMB promoter or EF1 alpha promoter. So, and the Cas9 construct is generally used. It's not different for each target. So it is a universal construct. And then the guide RNA sequence, the guide RNA is transcribed from the U6 promoter, and it has some different, it has the 20 sequence for complement each target and 80 nucleotides for the guide RNA backbone sequence.
So you say U6 promoter. What is it? Is it some kind of a plasmid, or what do you actually deliver to the cells?
The basic way is deliver the plasmid, but I prefer to deliver the protein RNA complex pre-assembled in vitro rather than the plasmid delivery.
Mm-hmm.
Yeah, because the ribonucleotide, the effect is more faster than plasmid because it doesn't require the transcription or translation time, and then it has less toxicity than the plasmid method because DNA itself, the plasmid itself, a little bit toxic to the cells. So I prefer the RNP delivery.
And so you work on just a cell culture, or do you ever work on like real tissues?
I'm working with cell culture cells.
Right. And so how do you make sure that they absorb this RNA protein complex? Can you just put it into the whatever medium and they'll just absorb it, or do you have to do something special for this to happen?
Usually using the electroporation.
And so what fraction of the cells are successfully affected by this CRISPR system? Is it close to 100% if you wait enough?
It very depends on the cell lines. Some cell lines uptake over 90% cells uptake the CRISPR construct, but some cell lines just less than 10% of them uptake the CRISPR construct. It very depends on the cell type.
What are your preferred cells? What type of cells work best with CRISPR?
For the plasmid delivery, the human HEC293 or HeLa cells are very good. And for the electroporation, the K562 or GM1287878 cell lines are very good for the later experiment.
And so for you, I understand this whole experiment where you perform the mutation and then sequence and look what the mutation was. So you can extract from this the information about how well different CRISPR systems work and maybe optimize them. Is that your main motivation?
Yeah, that's right.
But for others, they may want to screen these cells for a specific type of mutation which is desirable and then filter that out from all the other mutations. But then you have this problem that once you sequence the cells, you have to kill them. And so even if there was this good mutation among them, it is now gone. Those cells are dead. So how would you make sure in this scenario that you can trace that back? Like if you sequence and you know that the mutation you're looking for is there, how can you trace that back to some live cells that you can then continue to culture?
Okay. The experiment, the purpose is quite different. For the extracted DNA from the bulk cells or monitor the mutations in each cells. So if you want to verify the exactly wanted mutations in the cells, we have to make a clonal population from the bulk. and then analyze the mutations from each clonal population.
Right, but that wouldn't be very high throughput, right? Because
yeah,
you have to clone
yeah.
separately and then to sequence separately.
Yeah.
So if you're looking for a rare mutation, right, that would be hard.
Yeah, that's true.
So do you expect most users of CRISPRpic to have basically the same motivation of just looking for an improved CRISPR system? Or are there any other applications that you envision?
I don't like to restrict the purpose of our program for just monitor the mutations. Actually, our program is about analyze the mutations in the amplicon sequence.
Right. So that's an interesting aspect. So before you sequence, you amplify your region of interest. And what exactly is the goal of that step? Why do you amplify before sequencing?
Because the whole genome sequencing is quite expensive for if they want to sequence a lot. So if we sequence the genome from 1 million cells, we don't need to sequence all of the entire genome. So if we have a particular region of interest, we just need to sequence the target sequence only. So we can amplify. Amplify actually means the amplify the target sequence then compared to the whole genome sequence.
Right. So it's not that you need a lot more of the DNA material. You just use amplification to suppress all the rest that you don't care about.
Yeah, that's right.
And how big of a region do you amplify?
Usually it's decided by the Illumina platform. Usually we use the paired end with 75 read, but it can be extended to 150 paired end. Or if you use the PacBio or other sequencing platforms, you can amplify more long sequence. So you
amplify exactly the amount that you expect to be sequenced.
Yeah, around the sequence.
And then you probably, speaking about the costs of sequencing, you probably want to combine many different samples, or is your a single sequencing run is just many different cells with possibly different mutations, but still coming from the same experiment, or do you try to multiplex?
Yeah, multiplexing, it's good. So in our analysis, we showed that only 1,000 reads is enough to monitor the CRISPR efficiency. So for example, the one MySeq run can provide 25 million reads. So you can multiplex lots of sequencing libraries at the same time.
Yeah, but does that actually work in practice for you? Because you need some kind of barcodes, right? And they're usually much more limited.
It very depends on the experimental design. So if you want, for example, if you want mix hundreds of thousands sequence at the same time, you don't need to use the Illumina barcode. Just adding the random sequence in the sequencing primers, and then you can distinguish the libraries.
Oh, that's interesting. So you can just use primers as a barcoding technique.
Yeah, that's right.
Okay, and so then you have a lot of sequenced amplicons, and this is where CRISPRpic starts its share of work. Speaking of CRISPRpic, let's start with the name. So CRISPRpic stands for prefix index counting, and let's first define what you mean by the index here.
So CRISPR and experiments, you have target, right? No one's really do CRISPR without targeting, right? You have guide RNA in your mind. That's your experiment, right? That means you know where you're looking for or where you want to sequence. So that means, okay, we're looking at this region of interest in the genome by CRISPR-Cas9. And then we just look at that region, and then we can expect all the deletions and insertion and substitutions around that double-strand breakpoint, because we know where it will be. We're not just random sequencing, right? We expect the mutation induced by CRISPR-Cas9. So that's allow us to do pre-index most possible mutations at that site.
I think you use the word index in the paper several times, and it seems like maybe in some different meanings. So index here, you say, is just indexing the mutations.
Yeah, there's two different indexing, so it's actually confusing.
Okay.
Actually, so first one is it's hashtag, right? It's what we call maybe dictionary or a list of all the possible mutation. That's our first index. So we just get a list of all the possible mutations, right? And then we detect them by exact matching. But problem is we have already sequencing error as a background, right? So exact match will not work all the time. So it depends on your experiments. Sometimes 90% of sequencing read can match to our expected list of mutations with perfect match. But some experiments only 30% or 40% can match to one of our expected mutations. But that doesn't mean all reads come from wrong place. They contain some sequencing error someplace in their reads. That reason exact match cannot catch them, right? Because of that, actually alignment program actually developed to do some inexact match. But exact match is very fast, so that's the reason we try to stick to the exact match. Inexact match always costs way more time than exact match, but we cannot ignore the remaining reads cannot match to one of our expected mutation. So we move to the second step. So we're using camera short nucleotide sequence, let's say 8 base pairs, right? So we're using those 8 base pairs at the breakpoint, double-strand breakpoint, to the upstream and downstream. So we're moving outwards to the end. So since we only sliding 8 base pair, it's less likely contains that 8 base pair contains the sequencing error within them. So many 8 base pair index will not suffer from sequencing error. So that's the reason we can pinpoint what happening between these two indices, upstream 8 base pair, downstream 8 base pair, where we find them in the read. And then what is the relative distance between these two indices in the read compared to the expected distance on the reference amplicon we're looking for.
It's a good overview of how CRISPRpic works, I think. But which of these parts do you call prefixed index counting, and where does that name come from? I'm just curious.
So prefixed comes from all the mutations. So deletion, if any deletion induced by CRISPR-Cas9, they should encompass the double-strand breakpoint, right? So from there, you can expect, one base pair deletion towards the 5' at the breakpoint, or 2 base pair deletion towards the 5' from the breakpoint. And then you go all the way to the half of sequence, right? And then you can apply exact same thing to the downstream sequence, right? 1 base pair deletion at the downstream at the breakpoint, 2 base pair deletion, 3 base pair deletion, right? And so on. So we can get all the expected mutation if CRISPR-induced. Makes sense? So we just get all the possible deletions around the breakpoint, and then we can start insert nucleotide at each position, right? If breakpoint, we can insert A or C or G or T as one nucleotide insertion, or we can insert AA or AC, 16 different nucleotides at the breakpoints too, right? And so on. So you can have some expected mutation as a pre-indexed. Make sense?
I'm still not catching the prefixed part. What's prefixed here?
Prefixed means really prefixed.
Oh, prefixed.
Yeah, prefixed.
Okay.
Yeah, prefixed.
I thought it was related to prefix.
Oh, no. Prefixed. Yeah.
Okay. So it's prefixed index.
So we're looking only what we want to looking, because sequencing error, some mutation can occur outside of double-strand breakpoint, right? Maybe 20 base pair upstream of double-strand breakpoint. Maybe something caused that mutation or sequencing error. We're not sure, but they will not be, less likely they will induced by CRISPR, right? So we fixed on the mutation we're looking for that we believe that's induced by CRISPR-Cas9.
Right.
That's how we reduce the analysis time dramatically.
Right. So the first part, I think, of your algorithm is more or less straightforward. So the first part is you build this huge hash table of all the possible sort of simple or common mutations. And then you can look up the amplicon in that table and tell, if it's there, you already know how that sequence could have arisen from which mutation, right?
Yes, that's correct.
Right. And then if there is no match in the hash table, then you have to do something more complicated.
Yes, that's we call pattern matching using 8 base pair indices. That's second stage.
Right. So in the paper, you have this big and very scary, I have to say, flowchart, which describes the algorithm. But let's try to break it down, make it more accessible and less intimidating. Walk me through the algorithm if the amplicon is not in the hash table.
Okay, we try to find in the hash table. Okay, they're not in the hash table, and then we start looking. We have two different in this list of AIMers, 8 base pairs at the breakpoint. So upstream and the downstream, right?
You don't a priori know where the double-stranded break is.
No, we know. We should know because any CRISPR-Cas9, they have experimental design, right?
Right. So you know it in the reference, but not in your read. So in your read, you don't know.
No. So this index is actually built from your expected amplicons, right?
Right.
So and then that's starting from there. So we using our reference amplicon. If there's no mutation error, this is our baseline, right?
So the pattern matching part also relies on a pre-indexed data structure, right? It's not just the first part is you have this hash table, which is essentially an index, but for the second part, you also build an index.
Yes, from the reference. Yes, we actually stick to the prefixed space.
Right. Okay, so you in the reference part, in the reference amplicon, you know where the expected break site is. Yeah, and then what do you do next?
From there, so let's say breakpoint there, first 8 base pair to the upstream is your first index in the upstream list. And then let's say 1 base pair away towards the upstream, that's your second 8 base pair in the upstream list. And then 3rd base pair away and so on, right?
Right, so it's just a list of k-mers upstream of the break site.
By 1 base pair.
Yeah.
And then the order actually imply the location, the distance away from breakpoint, right? So first one is 0 base pair, second one is 1 base pair away, and then third one 2 base pair away, and so on, right? And then we build exact same thing to the downstream as well, right? So now that's our starting point. So we have some read not matched to one of our expected mutation due to some sequencing error somewhere in the read. So let's say we looking first 8 base pair from upstream. Can you find that in the read or not, right? So if we find that, right? So we found first 8 base pair in the upstream list. Okay, we find them and then we move to the downstream, right? We also found first 8 base pair in the downstream on the read. And then we're looking at is there any nucleotides between these two indices in the read? Maybe nothing. So they are not next to each other in the read. And then these are our first element from two lists. So they also not expected any nucleotide and the reference genome error, right? Because they are first from each set. So this is wild type. This read doesn't match to one of our expected mutation because they have sequencing error somewhere in the read. That's the reason they cannot match to the wild type sequence at the first place. But based on our pattern match, okay, we're looking at the first A base pair from both upstream and downstream list. We found them in the read. They are right next to each other. We conclude that these read are wild type. Make sense?
Right. So I think it's easier to explain the other way around. So if you have a deletion around the double-strand break site, around your expected break point, then you sort of look at the next k-mer to the right and the next k-mer to the left, and they in the resulting amplicon, they will be next to each other, right? So then if you start from the amplicon and you look at those two k-mers, then you can map them back essentially to the reference, and you will know how big of a part was deleted around the break site. The problem is that in your amplicon, you don't actually know, again, where the break point is. Instead of taking k-mers from the amplicon and mapping them to the reference, you do the other way around, because in the reference you know the break point, but in the amplicon you don't.
Yes, that's correct. That's the reason we're starting these k-mers in the read, and then their location actually imply where is the breakpoint. So, but sometimes this A base appear 2 times in the read, right? In that case, it's unclear. It's not clear, right? And then we throw them away because we cannot tell where will be the breakpoint among those 2 locations, right? For most case, we see only 1 time of this A base pair in the amplicon as well. And then, that's their breakpoint, around their breakpoint. But sometimes A base pair is kind of short. Sometimes you can see same A base pair 2 times in a sequence read amplicon. And then, we cannot tell among those 2 locations we found which one gonna be breakpoint. We cannot do that. So those things we discard them. But that's not that many cases, very few cases like that.
Right. So, I think it's just interesting because of this, right? Usually we think of mapping reads to the genome, but in your case, it's more like mapping genome to the read.
Yes.
Right? Because you take k-mers from the reference and you try to find them in the amplicon.
That's a good way to say. Maybe I need to use your way to saying to other people as well. That's a very good way to saying about our algorithm.
And so you essentially, in the reference, you go further and further apart from your expected breakpoint, and you continue until you start finding k-mers that occur in the amplicon, right? And that's how you know how much was deleted, or maybe something got inserted, and so on.
Yes, that's correct.
Yeah. I'm wondering what about like possible SNPs in your reference? So what if your reference does not correspond to the genotype of the cells you're working on? Does that ever happen?
So if that's the case, so this is our amplicon sequencing, right? So if there's nothing happen, and then there is SNP there, but it's wild type to that sample, right?
Yeah.
And then usually most abundant amplicon will be wild type. And then that will be our baseline, right? So if some person has a SNP in some person, but that's their wild type amplicon as the most abundant. So we can correct that at the beginning. Yeah.
Okay. So you don't actually use like the human genome as your reference, but instead you infer the reference from your reads.
Yeah. We cheating on the human reference genome as make sure we amplifying the reason we are interested in, right? At the same time, looking at our sequencing data, actually, we can see actually the SNP exists or not before starting analyzing data.
Okay, so you have to look both at the human genome, at the whole human genome, and at this most frequent amplicon. So does this happen inside your tool? How do you combine those two?
At the wild type sequence, already checking actually this is what we expect to see. So usually that's the reason our input takes as an input the expected amplicon and Cas9 breakpoint, right? So we're not providing this amplicon by ourselves. We ask the users to input that expected amplicon.
Okay, so it's on the user to extract that most frequent read and provide it to you?
Or they have to check. Yeah, they have to check.
Mm-hmm.
But they can start with reference genome, and then if that's the case, something will go wrong, right? You can tell easily, right?
Yeah.
Because wild type will not be most frequent at the final table, and then you will be aware of something wrong, and then maybe you can take a look at your data carefully, and then you provide expected amplicon considering this SNP position, and then rerun again. Since this takes only less than a minute, so you can run, I think, 2 or 3 more times to fix your error.
That makes sense. And then there are microhomologies. So can you explain what microhomologies are and why they cause problems?
The microhomology is a same repetitive sequence appear twice or more than one time in the amplicon. So because the microhomology sequence is the sequence are identical, so it can be aligned in many ways. So when the alignment program align the microhomology sequence inappropriately, and it is at the border of mutation region, is ambiguous. Sometimes it can be mutated or analyzed into mutated or unmutated. So it's a basic problem of the sequence alignment program. But in our program, the sequence hash tables already know the microhomology can be here and here, and we can distinguish this microhomology sequence mutated or not correctly.
Maybe I give you a simple example. So let's say A, we have 6 base pair deletion, right? But at the 6 base pair, first base pair is A, last base pair is also A, right? So that means you can make deletion A either first upstream or downstream. So usual alignment program, which A is deleted by chance, because to the scoring metric, scoring of alignment doesn't have any changes. If you delete the upstream A or downstream A, it's same penalty.
Right.
Make sense?
Yeah.
But to us, actually, we delete A and then upstream, downstream systematically, and then we know those both deletion end up same sequence.
Mm-hmm.
So we have prefixed index. We know, okay, these 2 deletion can end up same sequence, and then we annotate that in our hash table.
Right. So it results in an ambiguity in both your algorithm. Well, in your algorithm, it's not really an ambiguity, right? It's but you do say, like, I don't know what exactly the deletion was.
Yeah, there's no way you can do that unless you actually put the marker on each edge, right? And then there's no point of doing that anyway, right? So biologically, it doesn't really matter whether you delete upstream A or downstream A. Eventually, they end up same sequencing, right? So all we need to know is, okay, 2 deletion event can generate same sequence. That's all we need to know. That's the reason our deletion frequency by position is not biased at all. So when we see, okay, 10 amplicons match to this event, right? And then we know these 2 deletion events end up same sequencing. So we actually count 5 times for first deletion and then count 5 times for the second deletion, rather than picking randomly,
And so you're saying in a program based on alignment,
mm-hmm.
those might be split between multiple types of mutations or multiple different alignments, and they would be split randomly.
Split randomly, but within one alignment, they choose one side.
Yeah.
So sometimes, okay, you run program, this run, they pick all the upstreams only. And then you run second time again, maybe they pick all downstream. Within run, they're not actually picking up randomly, they usually put one direction based on some random seed in their program.
Yeah, and I just have to say that I don't approve of the word microhomology here because, homology is this concept from evolutionary biology, which means that 2 things have common evolutionary origin. But when you say microhomology, it just doesn't make any sense at all.
That's confusing too, but in the CRISPR Cas9 world, who Seung is expert in, they're using word that way.
Yeah.
I was so confused at the beginning, and then soon explained to me about that. And then, yeah, microhomology in the Cas9 CRISPR field, actually, that's the word for them. But to me, as a sequencing analysis person, yes, it's quite confusing.
Okay, I would like to say that it's not the word of CRISPR field. It's a word from the DNA repair studies. It's called the microhomology mediated end joining repair. So the microhomology is kind of the homology sequence between the upstream and downstream of the double-strand break site. Make sense?
Yeah, absolutely. And you're right that it sort of goes back to this, so originally there was this homology mediated repair, or what was it, like homologous end joining?
Yeah.
And so then they just made variations of that term, but of course that term was not very correct to begin with. But now we have it for too long to change, I suppose.
Yeah.
And so we talked about microhomology in the context of just sequence analysis, but this microhomology mediated DNA repair, does it itself matter in this context? Is it something you have to account for or to deal with when doing these sorts of experiments?
Usually there is some study shows the microhomology mediated repair is frequent than random deletion, random repair. I would like to recommend to consider the microhomology sequence between the upstream and downstream from the double-strand break site of the CRISPR have some advantage to get the mutations we wanted, such as the out-of-frame sequence.
Actually, in fact, beautifully, biological and mathematical say same thing. Let's say it's microhomology in there. So enzyme tend to connect them together, right? So you're going to see more insertion deletion between this microhomology, right? But at the same time, thanks to this microhomology, many different deletion insertion events ends up same sequence. So let's say our index, okay, this sequence has 5 different deletion mutation, right? So 5 different mutation can end up same sequence, right? Usually those the sequence have more frequent than any other sequence. So enzyme try to fix using microhomology to concatenate them, and then but deletion insertion happening at that event. At the same time, mathematically, microhomology actually ends up same sequence from different mutation events. So you see more actually frequent events in the sequencing count and at the same time. So our program actually, CRISPRpic is most beneficial when you target the area of region that has microhomology around it. Or other alignment program actually suffer from that, but ours are stand. So if you target some area that doesn't have any microhomology, our program and others are performance kind of similar. But when target region has microhomology, actually our program superior to any other approach.
Right, interesting. And so all the other options are based on alignment, right?
As I know, yes.
Right, but that's still not aligning to the whole human genome, but aligning just to the amplicon reference.
That's it. Yeah, because that saves a lot of time too, right? If you try to, you know where you're looking for, so they better to align to the expected amplicon rather than entire human genome, right? That way they can save anyway. So the Cas9 analyzer or CRISPR, they align back to the expected amplicon. But we do vice versa, right? As your expression, I like it a lot. We mapping reference to the read, right? That's what we're doing.
Right. And so you chose the K-mer size as 8, right? What were the considerations? What happens if you make it much bigger or much smaller than that?
That's one of the options. So we start 8 usually unique enough within 200 base pair, 300 base pair. Of course, if you're looking at the whole genome space, 8 base, it's very bad length. You have to be longer than 20 base pair. But since we're looking at the expected amplicon, which is about 200 base pair or so, right? usually 8 base pair tweak unique enough in most cases. But if not, actually, we suggest to increase 9 base pair or 10 base pair. But at the same time, so there's counter property, right? So it's like a sensitivity and specificity. So if you increase KMR, you have better chance of being specific, right? Unique in the space. At the same time, you have a higher chance of having sequencing error within that window.
Right.
So we're starting from 8, but you cannot find anything. Our program automatically increases to the 9 base pair and 10 base pair, 1 base pair incrementally to get the better result.
Yeah, so you have sort of auto detection of the KMR size?
So, let's say, as I mentioned earlier, this 8 base pair found in multiplex and the read amplicon, we cannot use them, right?
Yeah.
So when we build the list of upstream and downstream, so many indices actually found in multiplex, and then we actually automatically increase the 1 base pair to build a new list. Especially the 8 base pair cameras very close to the breakpoint within 3 base pair. That means first 3 cameras have to be unique within expected amplicon. Any one of them is not unique in the expected amplicon, we increase the 9 base pair camera instead of 8 base pair camera.
Okay, that's pretty cool. Is there anything else about CRISPRpic that you wanted to cover that we didn't talk about?
I think you covered very well. How about Seung Woo?
Yeah, I think so. But the most important thing for the general scientist is that the sequencing alignment program is take up very long hours or days to analyze 100,000 or millions of sequencing reads. But our program just take within 1 minute in the personal laptop, not the high computing servers. So yeah, I would like to advertise.
That's awesome. Yeah.
Good job.
So what are your future plans with this? So now you have this tool that works really well and it's very fast, as you say. Now I guess you'll move on to your actual problem, right? Of maybe designing a better CRISPR system. What will it be like? What are you going to do with this? How are you going to apply it?
Seung Woo, can you talk about the off-target plan or it's secret?
I don't like to say that, but using the CRISPRpic, we can study about each mutation. Or, for example, now the researchers using the CRISPR and knock out the genes and then just study the function of genes. But the CRISPR can induce a very diverse mutations in the targeted sequence. So not only the on or off manner, we also studied the function of each mutation. For example, not the genes. For example, the promoters or enhancers and transcription factor binding sequences. I think the CRISPRpic is very useful tool for analyze the mutation spectrum and mutation type from the very large sequence files. And then also the CRISPRpic is very useful for analyze the single cell data, not only the CRISPR-induced mutations, like the single cell barcode of the CRISPR mediated experiment or the perturb-seq experiment.
Oh, that's really interesting. So are you saying you could apply the single cell RNA sequencing methods to essentially DNA sequencing around your double-strand break?
Mm-hmm. Right.
That's very cool. So you essentially have this very high throughput barcoding at the level of single cells.
Yeah.
Have you already done an experiment like that?
Yeah, not very much, not very frequently, but yeah, I have experienced before.
Cool. there is also this paper, and you cited in your paper, so you're aware of it, that shows that when you have a CRISPR mutation, right, we usually look around this expected break site, but there are some large-scale mutations, some rearrangements, or they could be also like far away from the break point. And so I was wondering what your take on this is, and especially if you're designing a better CRISPR system, maybe your main concern shouldn't be what happens exactly at the break site, but what happens around it, because there could be much larger structural variations induced, and it doesn't look like CRISPRpic really can discern them, or in general this approach of using a short amplicon to classify the mutations.
Basically, the CRISPRpic can handle a very large sequence construct or structural variations, because whatever the sequence should be amplified by the polymerase and primers before sequencing. So the CRISPRpic just require the amplicon sequence, not exactly whole the amplicon sequence, because the CRISPRpic requires the end of sequence, and whatever the inside sequencing, the CRISPRpic can extract the sequence from the most abundant wild type sequence. So CRISPRpic can handle the a very large long sequence and any other structural variations. But the problem is the sequencing technology, and also it's true that the polymerase has a limitation of amplify the DNA stretch is up to 10 kbs. So I think it's a technical problem. The CRISPRpic can handle any issues you mentioned.
So are you saying you would amplify not just the region corresponding to the read length, but a much larger region?
Yeah.
Would that be the idea?
Oh, yeah, that's true.
So actually, CRISPRpic as a program doesn't have any limitation. As long as you provide good sequencing data, may contain these large deletions, right? But actually, the difficulty comes from actually sequencing itself. So as long as you provide good sequencing data to the CRISPRpic, we can handle that.
Right, but if you're still using short read sequencing, would it work with amplicons that are not all of the exact same region, but they are sort of pieces of a bigger amplified region? Would it still work correctly?
In that case, expect amplicon not just 200 SPL, right? It could be maybe 1 MB or 2 KB and so on. So in that case, we need free fixed index growing dramatically, right? At testing up to 2000 base pair, speed is not suffered that much, but I'm pretty sure we extend this length to the 100K or something like that. Yeah, my program could start going to be slow, I think.
But it would still work correctly if the read is much shorter than the whole amplicon.
Oh, yes. So expect sequence it's way bigger than what you see, right? So it's all based on your experimental design. So you have to put the primer outside of your deletions, right? Any primer within that deletion, large deletion, they're not even successfully amplified with anyway, right?
But you expect that the read is sort of flanked from both sides by primers, right? And it won't be the case in case of short reads and long amplicons.
These two steps, right? You have Cas9, CRISPR-Cas9 experiments, and then you wanna look at what happened by sequencing, right?
Mm-hmm.
So when you sequence, your primer will design, and primer will give you what kind of event you can see, right? So if a primer, any large deletion, any primer sitting on the large deletion, they cannot even amplify the read error. And then that's technical limitation, right?
Right.
That's what we meant. So as long as you have some better technical design, the sequencing design, and they have good sequencing data to detect this large deletion, afterwards, CRISPRpic can handle that. But if not, there's no way we can handle that. So you have come with very smart sequencing experiments to detect these large deletions. And then Seung Woo has some idea. We will work on that soon.
But I mean, if you have the two primers are far apart, and there's still like a significant deletions inside it.
In that case, we can delete that. We can detect them. Yes. So your primer is about 2 MB apart, right? 2 MB apart, and any deletion within that size, we should be able to see that.
Yeah, but that's a technical problem. So there's no polymerase can amplify the 2 MB.
Yes.
Okay, but even if it's much shorter, than that, but if the reads are even yet shorter,
maybe 2K, I think we can handle that.
Yeah,
easy without big problem.
So like 2KB amplicon, but if you're using like 200 base reads, would that work?
And then 100 base pair read will contain, let's say it's 2000 base pair deletion, right? And then for sequence will capture the planking outside of deletion, right?
Oh, right. So so you'll find that one sequence that covers the break. I see. Right.
Yeah.
Very nice. All right, guys. It was fun talking to you, and thank you for coming.
thank you.
Yeah, thank you for inviting.